Thank you very much for inviting me to share our work at the International Conference on Basic and Translational Cancer Research. Today, I'm going to talk about emerging nanotechnologies for cancer immunotherapy applications. Let me start by showing you a movie. Here, a cancer cell is getting attacked by an immune cell called T cell. And this is the future of cancer immunotherapy. Indeed, if you pick up any leading journal or a magazine, you'll see that they have highlighted cancer immunotherapy as something that is that has potential to cure cancer and indeed is a breakthrough. Now, despite this initial excitement, there are several challenges that were identified as more clinical trials went on and as we more understood how cancer immunotherapy works. And these challenges have hampered the effective use of cancer immunotherapy in curing the cancer for a variety of different types of cancer. My lab exactly works on addressing these challenges. Now, before I go into that, I would like to just give a brief overview of how our immune system works and how do tumors develop and what are the challenges that needs to be overcome in order to make immunotherapy as something that can potentially cure cancer. So our immune system is divided into two types. One is innate immune system and adaptive immune system. And there are different types of cells that are involved in launching a response in each type of immune system. For example, in innate immune systems, you have macrophages, natural killer cells, and antigen-presenting cells, whereas in adaptive immune system, you have T cells and B cells. Now, during an initial attack, for example, if there is a virus or pathogen that invades our body, innate immune system launches a rapid and non-specific immune response. For example, macrophages can eat up or phagocytose these foreign bodies. Natural killer cells can attack and kill the cells that have been infected by these foreign bodies, or antigen-presenting cells can either directly eat this foreign bodies or eat up the cells that have been infected. So there are multiple types of responses that have been shown that in this case, very rapid and non-specific response that takes care of any invading foreign bodies. Now, despite this first line of defense, the foreign bodies still grow. They overcome this first line of defense and still grow, and which is where you get infections. At this point, adaptive immune system is alerted and activated. Now, as I said, there are two types of immune cells that are involved in adaptive immune response. There are T cells and B cells. And we have a crosstalk that happens between innate immune system or innate immune cells and adaptive immune cells. For example, antigen-presenting cells, which generally present the antigens, which are specific markers on these foreign bodies, or cells that have been infected with these foreign bodies, such as pathogens and viruses. And these antigen-presenting cells then present those antigens to T cells or B cells. And then these T cells, which are activated, then seek out this and recognize the specific markers on the foreign bodies or infected cells and then eliminate them. B cells can release antibodies that can neutralize these foreign bodies, which again includes viruses or pathogens. So as you see that our immune system is very smart. It has evolved to target any potential threat, for example, any potential viruses or bacteria or pathogens, and learn to educate to launch the response. Now, despite being such a sophisticated response and having a smarter immune system, the question that comes is, how do tumors still grow? Tumors are smarter. They have evolved to kind of figure out a way to evade the immune response. They have several mechanisms that have been developed by the tumors that can evade the immune response. For example, tumors can release several cytokines and chemokines that can attract immune suppressive cells, such as regulatory T cells. They can dampen the immune response. They can also release immune suppressive factors that can modulate the functions of innate immune cells, such as macrophages, or in this case, as well as dendritic cells. Now, this macrophages, for example, not only not target the tumor cells, but actively help in the tumor growth. And we'll talk about that in next few slides. But tumors can release several immune suppressive factors that can modulate the functions of different types of immune cells. They can also overexpress several types of receptors that can bind to ligands on, for example, T cells, and hit the brakes on the activity of the T cells. For example, in this case, the T cells are activated and then ready to launch an attack on the tumor cells. But once they bind to these overexpressed receptors on the T cells, they know that they know that not to kill the tumor cells, which means that it acts as a checkpoint or a brake to target the tumor cells. Understanding this, there were several types of mechanisms. There were several therapies that were developed. For example, antibodies that can block these interactions, bind these receptors, and that can make sure that the brakes are released and the T cells can then target the tumor cells. Indeed, these types of antibody therapies have been shown to have a dramatic impact on several aggressive tumor types such as melanomas and non-small cell lung cancer and kidney cancer, and have been shown that it works very, very well for certain type of tumors. Indeed, several types of these therapies have already been approved for different types of indications that have shown to have impact in the clinics. Now, despite this initial success and the excitement about using the cancer immunotherapies that target several of these immunosuppressive mechanisms, there are several challenges that have been identified. For example, this immunotherapy, some of these blockbuster immunotherapies have been shown to have a low response rate. As an example, an immune checkpoint inhibitor antibody works in only 20% or so of the patient population in a variety of tumor types. Now, these therapies are very, very expensive. This shown to have high toxicity, whether you use as a monotherapy or you use in a combination therapies. Although there has been shown to have some impact in terms of increasing the response rate, the toxicity has been shown to be also high. And this can have a dramatic impact on the quality of life of patients. So in order to address these challenges, we are working on addressing two important questions. The first one is how can we improve the response so that as many patients as can respond to these therapies. And the second one is, can we design imaging probes that can allow us to monitor the response early on, that can allow us to stratify the patients that are responding and the patients that are not responding, so the patients that are not responding can be switched to an alternative, better approach or better therapies. So I'm going to talk about two different questions that we're trying to address as to, can we design nanoscale approaches that can allow us to target different types of cells in the tumor, immune cells in the tumor microenvironment, and that can allow us to modulate the immune response such a way that we go from immune suppressive environment to immune responsive environment that can allow us to have a significant anti-tumor immunity, and that can lead to tumor regression. And the second platform we try to focus on is to design, as I said, design imaging probes that can allow us to monitor the immunotherapy response early on. To go one step further, can we design imaging probes that can allow us to differentiate between resistant tumors and sensitive tumors, or responsive tumors, therapy responsive tumors versus therapy non-responsive tumors? And can we design that in a way that can allow us to identify much early on than current imaging probes? Today, I'm going to focus on designing nanoscale approaches to target macrophages that are in the tumor microenvironment, or called tumor-associated macrophages. And I hopefully will like to convince you that by targeting macrophages, we can effectively allow us to have a significant anti-tumor immunity that can lead to tumor regression or better anti-tumor immune response. The question that comes is why tumor-associated macrophages are important, and how do they help in tumor growth, and what approaches can we design to target these tumor-associated macrophages. Now, recent clinical trials have been shown that tumors have been are heavily infiltrated with these tumor-associated macrophages, and higher infiltration of these tumor-associated macrophages is associated with poor outcome in clinical trials, as well as including variety of different types of therapies. So how do macrophages help in the tumor growth, which is what, or help in tumor progression, which is what we'll talk about in the next slide. Now, macrophages are plastic cells that can exist in different phenotypes based on the environment they are exposed to. In tumor microenvironment, we can either find M1-like macrophages or M2-like macrophages. Now, M1-like macrophages are shown to have anti-tumor functions. They can, indirectly or indirectly lead to immune stimulation and lead to tumor suppression. Whereas M2-like macrophages have pro-tumorigenic functions that can lead to angiogenesis, tumor promotion, lead to metastasis, as well as directly or indirectly impaired T-cell immune response. Now, tumors, as they grow in aggressive tumors, you see that the M2-like macrophages are in predominant amount as quantity as compared to M1-like macrophages. So how does tumor recruit these macrophages? Tumors can release several chemokines and cytokines or chemoattractants that can recruit circulating monocytes to the tumor microenvironment. These monocytes, they can get differentiated into either M1-like phenotype or into M2-like phenotype based on the environment they're exposed to, based on the cytokines they're exposed to. In this case, if monocytes are exposed to GM-CSF or LPS or interferon gamma, they'll differentiate to predominantly M1-like phenotype. Whereas in case of higher levels of CSF1 or colony stimulating factor 1 cytokine or IL-4 or IL-10, the monocytes will differentiate it into M2-like phenotype. Now, it turns out that in tumors, there's a high level of CSF1 cytokine or IL-4 that can lead to a higher percentage of M2-like phenotype, which in turn lead to develop tumor progression and have pro-tumorigenic functions. Understanding and considering the importance of macrophages in the tumor growth, several types of approaches have been developed to target different types of nodes in the macrophages. For example, inhibiting the recruitment of monocytes to the tumor microenvironment, or inhibiting the differentiation of monocytes to M2-like phenotype, or depletion of M2 macrophages altogether in the tumor microenvironment, or repolarization of M2 macrophages to an M1-like macrophages. Now, it turns out that a better strategy, recent studies have shown that a better strategy would be to repolarize M2 macrophages to an M1 phenotype, because you don't really just deplete the macrophages, M2 macrophages, but you have M1-like macrophages that are endowed with these anti-tumor functions that can lead to tumor suppression, immune stimulation, and that can have long-term impact. So recent studies have shown that if you target colony stimulating factor 1 pathway, can lead to better repolarization of M2 macrophages to an M1 phenotype. So what is this CSF1R signaling pathway, and what are the different what are different strategies that have been developed to target these pathways? So CSF1 cytokine binds to this extracellular domain of CSF1R receptor that then dimerizes, and this dimerization lead to activation of signaling cascades, both extracellular as well as cytoplasmic domain, that can lead to phosphorylation of the intracellular residues, and this phosphorylation of the intracellular residues leads to activation of kinase signaling pathways that again lead to proliferation and survival of M2 macrophages. Now, considering the importance of this pathway in survival, proliferation, functions of M2 macrophages, several types of strategies have been developed. For example, antibodies that can deplete CSF1 cytokine, antibodies that can prevent the dimerization of CSF1R receptor, or intracellular tyrosine kinase inhibitors that can inhibit the phosphorylation of intracellular residues. Now, despite these several pathways, and even considering the importance of macrophages in tumor growth and progression, it turns out that majority of this inhibitors or monotherapies have not been effective in inducing significant anti-tumor growth inhibition. In this case, it's in a variety of different tumor models. Indeed, in our preliminary studies, we have shown that one of the most potent CSF1R kinase inhibitor, intracellular kinase inhibitor, works for only 5 minutes. So, translating this into clinical trials, also recent studies, clinical trials have shown that small molecule CSF1R inhibitors are not really effective in in getting an overall and getting a significant overall response rate or inhibiting the tumor growth as monotherapies. So the question that comes is, we know that macrophages play a very important role in tumor growth. They are in a significant amount in the tumor microenvironment, especially in advanced tumors. And CSF1R pathway play a very important role in macrophages and tumor-associated macrophages growth, as well as proliferation and functions. So why are these therapies not working? What can we do to make to increase the impact of this or effect of this anti-tumor of this tumor-associated macrophage therapies? So we asked ourselves several questions in order to understand what can we do to what are the important factors that can allow us to enhance the effectiveness of these therapies and increase the anti-tumor effect. The first question is sustained inhibition of C7R pathway important? Now, why is that important? Because recent studies have shown that sustained inhibition of kinase pathway is needed for improved therapeutic activity. We have recently shown that if you have if you inhibit the PI3K pathway in cancer cells in a sustained manner, that can lead to significant anti-cancer effect. So based on the same principle, we wanted to try and see if the same sustained inhibition of CSF1R kinase pathway in tumor-associated macrophages would lead to better anti-tumor effect. The second question is sustained inhibition of downstream signaling pathways or feedback signaling pathways important for getting for better repolarization of macrophages? In this case, downstream signaling pathways of CSF1R signaling, does it lead to better repolarization efficiency? Is enhanced repolarization efficiency required? For example, can we get better repolarization of M2 to M1 macrophages by inhibition of CSF1R and downstream signaling pathway, and does that lead to better anti-tumor effect? And then, in order to achieve all of this, do we need higher intracellular concentration and higher intratumor accumulation of the drug? And this is one of the biggest challenge in has been one of the biggest challenge in translating some of these inhibitors in the clinics, where having an inhibitor in the macrophages that can inhibit this signaling for longer period of time, a sustained inhibition of this signaling pathway, that can lead to better anti-tumor effect. Now, in order to kind of address these challenges of higher intracellular concentration and intratumoral accumulation, we wanted to design a system, a nanoparticle system, that can allow us to deliver this inhibitor in CSF1R inhibitor in the tumor microenvironment. It turns out a lot of recent studies have shown that the tumor vessels are leaky, and this leaky tumor vessels, because of this leakiness, nanoparticles can preferentially accumulate in the tumor, especially in tumor-associated macrophages, as compared to normal tissues. So with that, we wanted to the hypothesis was, can we design nanoparticle system that can preferentially deliver a CSF1R emitter to the tumor and effectively switch the M2 macrophages to an M1 phenotype that can in turn lead to better anti-tumor immune response and better anti-tumor efficacy. So you want to design a simple but effective nanoparticle system that can allow us to deliver this inhibitor into tumor-associated macrophages in the tumor microenvironment. So we started with conventional liposomal systems, where we wanted to design, encapsulate the CSF1R emitter in the lipid bilayer systems. But unfortunately, we saw that the nanoparticle was liposomal system was significantly unstable, with precipitation observed in less than 1 hour of synthesis. We saw that the loading was very, very low. We could not load more than 2 mole % of the drug in the nanoparticle system. So, thinking about translating this into a next stage in animal trials to kind of get see if it actually works in the system, we wanted to design something that can allow us to have a very high concentration of drug loaded into the nanoparticle systems. We don't have to inject a lot, and that can lead to better accumulation in the tumor. So indeed, the stability of nanoparticles at the drug concentrations that are again injectable, that are required for clinical efficacy, has been one of the biggest challenge in clinical translation of nanomedicines. So in order to address this challenge, we came up with a novel approach we call supramolecular nanoparticles. Think about this as a Lego system. So where we choose any molecular building blocks. Could this be lipids, polymers, or peptides, proteins, or antibodies, or small molecule drugs? And we use these molecular building blocks, and these molecular building blocks, and they can form a self-assembly, a complex structure, three-dimensional structure formed by weak interactions, hydrophobic or hydrophilic or van der Waals interactions. And based on the kind of molecular building blocks you pick, you can get a complex structures. You have a multiple systems where you have multiple building blocks that can form a three-dimensional system. One of the biggest question was, can we design a system, something like that, where we can have a CSF1R that is encapsulated inside the drug, inside the lipid bilayer system, and then it can form a stable nanoparticle system with high drug loading. One of the biggest question that is what governs these interactions, what governs this supramolecular assembly, and how can we design system by understanding those interactions? So in order to answer those questions, we wanted to see, can we step back and kind of figure out a way to see what interactions are happening and what makes the drug encapsulation unstable in a lipid-based systems, and can we understand those interaction in order to design a supramolecular system that can allow us to have high drug loading, high stability, which will in turn lead to better accumulation in the tumors. To do that, we wanted to use computer simulations, because computer simulations have been widely used for understanding drug-lipid interactions, for designing drug molecules, and as a surrogate for cell membrane that can allow us to design a better drug that can interact with the cells and then internalize in the cells, but has never been used to design a lipid-based nanoparticle systems that can allow us to kind of understand the interaction of the drug, the lipid bilayer systems, and then make a stable liposomal nanoparticles, so supramolecular systems. Indeed, computer simulations have been used for understanding atomic level details of biomolecular interactions that are not possible with any current experimental techniques. You can get the spatial and temporal resolution of the interactions of different molecules that are interacting at different spatial order and in a temporal order that you cannot really get using any experimental techniques. For example, all you see in when you're making a nanoparticles, all you see in a round bottom flask could be something that is precipitating out if you're thinking about self-assembly systems. So we wanted to kind of use this platform to understand the interaction first, and then design a system to make it stable. So have a predictive model of why drug is not making the system stable at high enough concentration, and what can we do to overcome those interactions. So just give an example. We develop this computer simulation algorithm where we start with quantum mechanical structure optimization of a parent drug, in this case any kind of drug that we are interested in, and then we do an all atomistic simulation of self-assembly, where this is a model lipid bilayer systems. Have the gray layer is a hydrophobic part of the lipid bilayer. The hydrophilic part is in orange and blue spheres, and then the drug is in the red, and we see how the drug is interacting and making it unstable. We do automated analysis of the stability, and if we see that the drug is at the concentration that we are interested in is making the lipid bilayer very unstable, we do we understand the interactions and then do a structural modifications to the drug in a way that it can stabilize those interactions and make the lipid bilayer system very stable. And once we identify and predict the structure, we then experimentally validate to see that whether it actually forms the supramolecular nanoparticle system or not. Just to give an example, we'll start with paclitaxel. Paclitaxel has been very widely used drug for a variety of different types of cancer indications, but it is a bulky molecule, has been shown to be notorious to form a stable lipid bilayer system at high drug loading. For example, it cannot load. It has been very hard to load more than 5 mole% of the paclitaxel in lipid bilayer systems. So we started with paclitaxel, and we did computer simulations to see that the paclitaxel kind of interacts at the interface. On the contrary belief, where it was kind of believed that it likes to interact with the hydrophobic part, it actually likes to interact at the interface. It forms large aggregates at the lipid bilayer interface, and makes the lipid bilayer very unstable. At high enough concentration, in this case, you have 20 mole% of the paclitaxel in the lipid bilayer system. When we did structural iterations, just by modifying the drug with an anchor, in this case, we conjugated the cholesterol, we see that it still makes it unstable, relatively stable, but still unstable at a concentration that is required for making the stable system. But when we see that, we thought that just having maybe not just the drug structure or the anchor, but also the linker plays a very important role. So we did a next iteration where we identify another molecule, and when we ran the simulation with this, we see that it indeed forms a very stable system. In this case, the drug is very well anchored, where the hydrophobic part of the tail system gets very well anchored and interacts very well with the hydrophobic part, whereas the drug interacts at the lipid bilayer interface, which is what it likes to interact with, and it forms a very stable lipid bilayer system. So we see that not only structure of the drug or the lipid anchor, but the linker also plays a very important role. So all components come together. Once we identify these components, it can make a very stable lipid bilayer system at much higher concentration. They show here we start with paclitaxel, and you see that it forms it precipitates out very early, and you see that there's a significant lipid bilayer disruption that happens. Whereas when we have a computationally identified structure and we actually experimentally validate it, we can achieve 6 times more loading, and it forms a very stable lipid bilayer system. So we have, the interesting aspect about this platform is you can adapt it for a variety of different types of drugs. So when we have shown it for paclitaxel, we wanted to see, can we actually adapt it for other types of drugs? So we started with PI103. Now, PI103 is one of the most potent PI3K inhibitor. It is planar molecule, but again, it has been shown to kind of disrupt the lipid bilayer system. It cannot load more than 2 mole% of the drug in the lipid bilayer system. As you can see here, it forms a large aggregates, but interestingly, it forms aggregates where it forms a pi-pi stacking. As you can see here, it forms large pi-pi stacking, forms these large aggregates that kind of destabilize the lipid bilayer systems and make it very unstable. But again, when we and again, to show that this is a pi-pi stacking that happens. So when we identify a molecule using computer simulations, and when we run the simulations, we see that it indeed forms a very stable lipid bilayer system, very stable supramolecular nanoparticles at much higher concentration than the free drug. So we can achieve a significant higher loading with significant stability just by doing a slight modifications to the drug. And beauty of this system is we can not only make it stable, but we can also identify different types of interactions that are happening, whether it's drug-drug interactions or drug-lipid interactions. And by understanding those interactions, we can design a system that can make it much more stable. We have shown that we can use this computational platform to understand drug-lipid bilayer interactions at molecular levels that was never been possible before, and we have adapted this platform for design of supramolecular nanoparticles. We have used it for a variety of different types of drugs, inhibitors, including chemotherapeutics, immunomodulatory agents, epigenetic drugs, and can be used for a variety of different types of lipid excipients. So now, going back to a system, now we wanted to use this platform to design a supramolecular nanoparticle systems that can allow us to deliver higher concentration of CSF1 inhibitor to the tumor. So the idea was, can we design the system that can allow us to, and using this computational platform, can we design a system that can allow us to address all these challenges? So we started with BLZ945, is one of the most potent and most selective CSF1R inhibitor, but never made it to the clinical trials because of the less efficacy that has been shown in early studies. but it is a very potent drug. So we wanted to start with this with this drug and kind of use the same platform and to see how it, at a higher concentration, what interactions are happening and how does it make unstable lipid bilayer systems. As we've seen in previous models, we see that even BLG945 kind of likes to interact with the lipid bilayer systems. It makes the lipid bilayer unstable by forming large aggregates at the interface. But when we identify, a structure by using simulations, we see that we get much stable lipid bilayer systems at much higher concentrations. So again, validating it experimentally as well as theoretically using computer simulations. So we designed and synthesized this molecule and synthesize the supramolecular nanoparticles, and we see that we get nanoparticles of around 100 nanometer size. They are stable using TM. They're stable for longer period of time, as long as 30 days, using storage conditions. But they release drug in a sustained manner in cell lysate, and we get much higher drug accumulation inside the tumor, inside the macrophages for longer period of time. In this case, as long as 18 hours. So we do get higher accumulation as well as as well as a longer accumulation of the drug in the tumor-associated macrophages. Now we wanted to see, does this stable system, which can release the drug in a sustained manner and can accumulate into the macrophages for a longer period of time, leads to sustained inhibition of CSF1R signaling. Indeed, we see that when we use when we perform the inhibition, perform the assay for understanding the CSF1R phosphorylation inhibition, we indeed see that we get the sustained inhibition of the CSF1R pathway as long as 72 hours. When we use the nanoparticle system, we see that we get sustained inhibition for longer period of time, whereas the free drug only works for 5 minutes. Indeed, this is very significant because the first time anybody has shown that by designing a system that can release a drug in a sustained manner and have higher intracellular accumulation, you can get sustained inhibition of this CSF1R signaling pathway for longer period of time, as long as 72 hours. The question was, does this sustained inhibition lead to inhibition of downstream signaling pathways as well? The question was, what are the downstream signaling pathways for CSF1R, and then do we see an effect on that? So we used a string map to kind of identify what are the pathways that are linked, that are directly downstream of CSF1R. And when we did a Western blot, we indeed see that we are getting sustained inhibition of CSF1R pathway as compared to BLG945, which is the parent drug, and then PLX3397, which is again one of another potent CSF1R inhibitor, not selective but potent CSF1R. It also inhibits other signaling pathways. But we do see that we can get downstream signaling inhibition of PI3K, which is downstream of CSF1R. We get higher SOX3 to SOX1 ratio, and then we get inhibition of arginase. So these studies have shown that we're not only inhibiting the CSF1R pathway, but we are also inhibiting the downstream signaling pathways that are important for M1 macrophages, and activate the pathways that are important for M1 signaling, and inhibit the pathways that are important for M2 signaling pathways. So, we see that by having a sustained inhibition and inhibition of downstream signaling pathways, we are kind of tipping the balance from M2 to M1. So in order to kind of validate these results, we ran a flow cytometry assay where we treat the macrophages to IL-4 treatment, which are shown to repolarize macrophages to M2 phenotype. And then we do the drug treatments and replace the media and then collect the samples for flow cytometry. We have used two different types of markers. One, CD2-Noxis, which has been shown to be a marker for M2 macrophages. And then our MSC2 and CD80 are shown to be markers for M1 macrophages. And then CD11B is a pan macrophage marker. And we did three different treatments: vehicle, free drug, as well as the supramolecular nanoparticles. And then we see that with the vehicle and free drug, we do not see much decrease in the CD2-Noxis expression, but In case of supramolecular nanoparticles, we see a significant inhibition of CD206 expression and subsequent higher increase of M1 markers as compared to vehicle or BLG. In this case, indeed, we see that if you compare M1 to M2 ratio based on the surface markers, we get much higher M1 macrophage in case of SN supramolecular nanoparticle treatment as compared to M2 macrophages for long period of time. And really exciting results, because it's the first time we have shown that by inhibition of CSF1R signaling pathways and as well as downstream signaling pathways, you can repolarize macrophages. As long as 72 hours, we can get much higher M1 to M2 ratio. The question was, does it actually translate to better anti-tumor efficacy? The first question is, can we actually get enough drug into the tumor by using supramolecular nanoparticles? Indeed, we see that we get much higher accumulation in the tumor. This is, we used NIR dye-tag nanoparticles, where we see that we get much higher accumulation in the tumor as the time after the injection, as the time goes, as compared to other organs. And we then studied its efficacy in two aggressive tumor models, in this case melanoma and metastatic breast cancer, and we see that we get significant tumor growth inhibition using supramolecular nanoparticles as compared to BLG or as compared to anti-CSF1R antibody in both melanoma as well as metastatic breast cancer. Let me stress a point that these SNPs are not targeting tumor cells directly, but they are targeting tumor-associated macrophages that can have a significant impact on tumor growth. They're not toxic, as we have seen. We have also performed additional tests, but just to show that there's not much change in tumor growth, we don't see any systemic toxicities. We see that there is a complete tumor metastatic inhibition with no metastatic nodules observed in the lungs. We see that, as expected, much higher M1 to M2 population. We see that we get much higher M1 macrophages in SNP treated tumors as compared to vehicle or BLG treatment. And we see that there's a significant survival advantage when we have SNP treatments, in this case metastatic breast cancer model, as compared to BLG or vehicle treatment. So just to summarize this section, we have shown that by designing a computationally inspired supramolecular nanoparticle systems that can have a higher drug loading and higher stability and have a sustained release of the drug and higher accumulation in the cells, we can have a sustained inhibition of CSF1R signaling in tumor-associated macrophages that can lead to significant inhibition of downstream signaling pathways, and that leads to enhanced repolarization of M2 to M1 macrophages that again lead to better anti-tumor effect because of the higher intratumoral accumulation. So we see that we can get the CSF1R inhibiting supramolecular nanoparticles to have significant tumor growth inhibition. It also inhibits metastasis and improves survival. So I have shown here that we can design system nanoscale approaches, in this case using computer simulations to design better nanoparticle systems that can allow us to target different types of immune cells in the tumor microenvironment that can lead to enhanced anti-tumor immune response, and that can lead to tumor regression and increase in survival. With that, I would like to acknowledge my outstanding students who perform all the studies, my collaborators who have been very helpful in a lot of studies that we have done, and the funding agencies in trusting our work. And thank you all for inviting us to present our work. Thank you.